Welcome everyone. We're going to continue the Josep Carreras Research Institute Distinguished Lectures in this Friday before holidays next week. So it's really a great pleasure to have with us Dr. Anthony Letai. Dr. Letai graduated from Princeton University with AB in physics. Then he received his MD and PhD at the University of Chicago and a complete clinical training in pathology and oncology at Dana-Farber Cancer Institute. He was a postdoc with Stanley Korsmeyer, and since 2004, he has run his own lab at the Harvard Medical School and Dana-Farber Cancer Institute. His work has been seminal in understanding the mechanisms of apoptosis and how cancer cells escape from apoptosis. In this regard, beyond developing assays like BHD profiling, he has been also critical in development of antagonists for BCL2, BCL-XL, and MSL1 that are, some of them are used now in the clinical setting. Among these, I will say that one of the most successful stories has been venetoclax, that now is widely used. He has also many roles in science and also the president of the Society of Functional Precision Medicine. We believe in personalized medicine. I think that this is a great idea. And he has been recognized by several awards, that the European Cell Death Organization Career Award, the Smith Family Prize for outstanding scientific contributions, and the Institute Outstanding Investigator Award. So it's a pleasure to have you here. The title of his talk is Guiding Drug and Cell-Based Cancer Therapy with Mitochondria.
Thanks, Manuel. It's a true pleasure to be here and to finally visit your institute here. Let me make sure I get the controls right. We'll start disclosing some of my extracurricular activities. I'm not going to talk about any of the drugs from any of these companies that I work with, but I will be talking a lot about BH3 profiling, and my employer, Dana-Farber, has all the patents on BH3 profiling. So I'm going to start with just a clinical case that stuck with me from way back when I was a fellow, and then I'll circle back to that. But this is the sort of problem that provoked a lot of the work that I did in the subsequent 20 years. I know for a lot of time, I'm sort of a physician scientist. I did an MD and a PhD, and for a long time, I struggled to find a connection between my medical work and the research that I was doing. Sometimes I felt that there was no more cohesiveness between my medical work and my scientific work than if I was sort of a ballet dancer and a plumber. There just was no real connection that I could find. But eventually I found one. And it kind of starts with this patient. So this is an all too typical problem. This is around the year 2000, over 20 years ago. AML, despite what we see in the hospital, there's a real selection for younger patients for AML in the hospital, because the older ones never even get to the hospital because they can't tolerate the sort of treatments that we give them, the harsh induction therapies. So an all too common issue would be the AML is largely, though, a disease. Most people are diagnosed over the age of 65 with AML. And most of them are unfit or wouldn't be able to tolerate the harsh induction therapies that we give them that require a long hospitalization and very intensive chemotherapy. And this was such a man. He had serious heart problems, what we call unstable angina, at the same time that he had this diagnosis of AML. And it was very frustrating because here it was sitting across the table to a living, speaking, completely intact person with this fatal disease, and there was next to nothing we could do for him. He died within 2 months. He was put on hydroxyurea that gave him some very temporary benefit. And then he died. And it was a frustrating case that stuck in my mind because AML, acute myelogenous leukemia, is a disease of older people. And for most of them, we couldn't really do anything that effective. So I'm going to get around to how we might benefit a patient like this, I'm going to address some very fundamental questions that I'm surprised aren't asked more. And one is, why does chemotherapy ever work? I was talking about how we couldn't find something for this guy, but actually, it's an absolute miracle that chemotherapy ever works to cure cancer, and we do cure some people of cancer. And I'm going to describe how I think an important answer to that question involves mitochondrial apoptotic priming, and I'll explain what that is, and also how we can use some of those principles to do a better job of precisely choosing drugs for our patients. So why should we care about conventional chemotherapy? When I say conventional chemotherapy, I mean those old-fashioned kinds of chemotherapy like Cytoxan and Adriamycin. These are the types that people we're supposed to be getting away from with our targeted therapies because they make people's hair fall out, makes them nauseous, lowers their white blood counts. It's the bad kind of chemotherapy. But you know what this kind of chemotherapy does? It cures people of cancer. It cures people of cancer in a way that none of our targeted therapies really can do. There's some magic there that we don't understand, and they're essentially the only drugs that we give that can actually cure people. And by cure, I mean the patient comes to you with a fatal disease, you give them the medicine, and the disease goes away forever. That's a really rare event, and almost the only drugs that we have that can do this are the old-fashioned, dirty, nasty chemotherapies. So just to give you an idea, in a month, if you want to compare the number of patients worldwide who are cured of cancer by conventional chemotherapy, it would be something like the number of people who could fill this stadium. If you want to look at the number of patients who are cured of cancer every month by small molecule targeted therapies like your kinase inhibitors, and I mean not just treated, I mean cured, it's very few. This happens to be a little town in Montana that I did a fourth-year clerkship in. Obviously not a lot of people. And my point is, although the cancer cell papers, the cancer discovery papers, the grants we write are all about detailed mechanisms and how to target them very narrowly with really highly advanced modern molecules, almost all the cures we've ever observed in medical oncology, and there have been tens of millions of them now, largely in lymphoma, leukemia, and testicular cancer, almost all these cures take place due to conventional chemotherapy. Now, these conventional chemotherapies largely target DNA and microtubules. DNA and microtubules are present in every single cell in our bodies, so it is not immediately evident why these drugs should be selectively toxic to cancer cells. So I'm going to address three big questions, I think, in chemotherapy. One, why does it ever work? Okay, something that's been observed for a long time in cancer treatment is that almost always solid tumors do worse than liquid tumors. That is, there's no solid tumors, no advanced solid tumors really that we can cure with chemotherapy, whereas we can cure some leukemias and lymphomas with chemotherapy. Also, there's this question that we don't get around too much, but it's actually the big problem in conventional, in oncology treatment, and that is when we take a patient and treat them with one drug and then they relapse, they're not simply resistant to that drug or couple of drugs that we gave them initially. They tend to be pan-resistant, and that becomes a huge clinical problem, this pan-resistance. But we actually don't have a very good understanding of what underlies this type of pan-resistance, and I'm going to try to offer you an explanation. So I'm going to get back to answering these questions, but I just need to introduce the mitochondrial pathway of apoptosis so that everyone in the audience is familiar with it. And I won't get too much into the details here, but it's key to understand that the mitochondrial pathway of apoptosis is regulated by the mitochondria. And the key event that determines or commits a cell to apoptosis via this pathway is permeabilization of the mitochondrial outer membrane. When the mitochondrial outer membrane permeabilizes, there's a loss of the transmembrane potentials inside the mitochondrion, and there's also a release or an egress of intermembrane space proteins. And both of these events are important because we can use them to monitor permeabilization of the mitochondria, and that becomes important in some of our methods. This permeabilization event is regulated by the BCL2 family of proteins, and it includes pro-apoptotic proteins like BAX and BAC. So BAX and BAC are absolutely essential because they actually form the pores. They homo-oligomerize, and they form the pores in the mitochondrial outer membrane. There's proteins that are also pro-apoptotic, like BID and BIM, that stimulate BAX and BAC to homo-oligomerize. Then the anti-apoptotic proteins that you've probably heard of some of, like BCL2 is the founding member, that's what the whole family is named after, but it has cousins, BCL-XL and MCL1, for example, and these are all anti-apoptotic, and they all basically work the same way. These anti-apoptotic proteins have in them a hydrophobic groove, and they're able to bind and sequester pro-apoptotic proteins to prevent their interaction and prevent their homo-oligomerization so you don't get these pores formed. So this hydrophobic groove is bound by something called a BH3 domain that's present in the pro-apoptotic proteins. These BH3 domains are amphipathic alpha helices, about 20 amino acids in length, that have a hydrophobic face that binds into that groove. And one of the things I found when I was in Stan Korsmeyer's laboratory many years ago was that we could simply synthesize these BH3 domains, these BH3 peptides, synthesize these 20 amino acid oligomers, and they could largely reproduce the activity of the pro-apoptotic molecules. So if you consider that every cell in your body, or every cell really, is in some sort of balance between these pro and anti-apoptotic proteins, what we could do is titrate in the BH3 peptides and determine how much of that BH3 peptide do we need to sort of like favor the pro-apoptotic side of the equation until the mitochondria permeabilize. And that's what we eventually began to call BH3 profiling. BH3 profiling is just the systematic addition of BH3 peptides of known concentration onto mitochondria and figuring out how much mitochondrial permeabilization do you get for a given unit of BH3 peptide. And our general readouts, like I said, are either cytochrome C release or loss of transmembrane potential. I'll be talking mainly about cytochrome C release today, but it's just a readout. If you need to add a lot of BH3 peptide in order to cause permeabilization, well, that's a cell that's relatively far from the apoptotic threshold, what we call relatively unprimed for apoptosis. But if you only need to add a little bit of peptide in order to induce permeabilization, that is a peptide that's very close to the threshold of apoptosis, what we call very primed for apoptosis. We do this two main different ways, and it's a matter of convenience. It largely depends on whether we have an adherent cell or a non-adherent cell. So for non-adherent cells, for leukemias, and I know this is founded as a leukemia center, we do our studies mainly by flow cytometry, where we take a single cell suspension. In order to get the peptides into contact the mitochondria, we gently permeabilize the plasma membrane with digitonin. Then we add the BH3 peptides. We incubate for about 60 to 90 minutes, and then we measure whether or not there's cytochrome C release, which is our measure of mitochondrial outer membrane permeabilization. So you'll see a lot of graphs where I'll show you where the y-axis will be some index of what we call priming or loss of cytochrome C. And you can see it's very easy in a flow cytometer to resolve cells, a population of cells that has cytochrome C intact versus a population of cells that have gone through this treatment process with one of our peptides, the BIM peptide. You can see there's an easy signal to read there. We do something very analogous with solid tumors with automated microscopy in 384 well plates, where we plate out the cells, permeabilize them, add the peptides, and then read out cytochrome C release using a bunch of automated microscopy codes that we train the microscope to recognize individual cells and train it to detect whether or not there's cytochrome C in the vicinity of the nucleus of these individual cells. So both of these actually give us single cell information, and sometimes we use it as single cell information, sometimes we aggregate it for bulk findings. The results and the information we get out of this really depends on the particular peptides that we use. And I'll start by talking about these promiscuous peptides. So promiscuous peptides measure net anti-apoptotic reserve. What you see here is a binding chart where on the y-axis you see the anti-apoptotic proteins like BCL2 or MCL1 and so forth. And on the sort of x-axis across the top are the names of the different BH3 peptides we used, and they take the names of the proteins from which they're originally derived. And this binding chart tells you which BH3 peptides interact with which proteins. So BIM, BIT, and PUMA are notable that they interact with all of the anti-apoptotic proteins, and therefore they can give you a measure of the net anti-apoptotic reserve, or these are the ones that are really good for telling us how close a cell is to the edge, how close it is to the threshold of apoptosis. So just to conclude this idea of apoptotic priming, when something is really close to the edge, we describe it as highly primed for apoptosis, highly primed for death, sort of like the individual who is sitting in the cab of that pickup truck there. Whereas ones that are further from the edge, we describe as being as unprimed or less primed for apoptosis, sort of like the people who are further from the edge and obviously at less risk for any sort of bad outcome. So let's get back to this question. Why is there a therapeutic index for drugs that target DNA and microtubules? And just to remind you the sort of magnitude of this index, let's say you cure someone of leukemia. Well, when they presented to you with leukemia, they had something like 10 to the 12th, 10 to the 13th leukemic cells in their body. So that's 12 to 13 logs of kill you need to accomplish with your drugs to cure that person. There is not a single vital organ in your body that can withstand even a single log of kill. that would be a 90% reduction in the mass. Can you imagine if you lost 90% of your heart, your lungs, your liver, your brain? You would die, obviously. So there's an amazing therapeutic index, and it actually surprises me that there isn't more study of this, because after all, I just want to emphasize this, this is the stuff that actually cures people of cancer. And yet I don't think we have a great idea for what causes this, what is the basis of this therapeutic index. Now, I think if any of us have ever asked this question, like I did early on in my training, what you're told is that, oh, it's because cancer cells are dividing really fast and normal cells aren't, and that's why you have this great therapeutic index. I think you would be surprised if you do what I did and actually engage with the literature and try to find any data to support this contention. It is astonishing that this is the principle supposedly underlying the only thing we have that cures people, and you don't see any evidence of it in the literature. Look at the literature for there is some early evidence from like 40 years ago when people were using people like Dana-Farber were using anti-metabolites, and they found that cells that were metabolizing very rapidly were more sensitive to anti-metabolites. But actually the clinical measurement of proliferation rate and its correlation with outcome after chemotherapy treatment, just find me a paper that's systematic. It's very hard to find any evidence to support this. And this is an example. This is the first of several pieces of data that I'm going to share with you that's unpublished data from Chris Sorosik's lab. Chris, and it's done by him and Cameron Fraser, one of his technicians. Chris was a postdoc in my lab, and he started this work many years ago in my lab, and now is really leading it in his laboratory. But what he did, just as like a simple method, is he used online databases to look at one index of proliferation, which is the transcript levels of PCNA, proliferating cell nuclear antigen. And he compared that to like basically how long people lived. And what he found is across all of these different diseases, there's next to no correlation between how rapidly something is dividing and how well the patient did. And I'll also just ask any of you who have clinical exposure, there's so many counterexamples of slow dividing things that are very sensitive to chemotherapy. A great example, actually, are the indolent lymphomas. Indolent lymphomas start out very sensitive to chemotherapy in utero. Usually it's pretty easy to get several logs kill of an indolent lymphoma. Hard to cure them, but you can still kill many logs of those lymphomas. What sometimes happens with these lymphomas is they transform into an aggressive, rapidly dividing lymphoma. Well, I guess that should be really chemosensitive, right? We experience exactly the opposite. These transformations into aggressive, rapidly dividing lymphomas are generally much less chemosensitive than the initiating indolent tumor from which it arose. So I would just put to you that, like, if you feel like proliferation is already a well-established fundamental biological explanation for why chemotherapy works, I would just say it remains to be proven, and I'm still waiting to see the proof of that. So I think there must be other things that contribute to this. And of course, I'm going to describe what I think it is. I think that it might have to do with how primed the cells are for apoptosis. And the idea is this: pretty much all cells, if you damage their DNA with chemicals or damage their microtubules with chemicals, almost all cells endure some kind of apoptotic or death signaling. However, if they start really close to the edge, what we call a primed cell, if you give them chemotherapy and they induce some death signaling, they're very close to the edge and they commit to apoptosis, because apoptosis is a switch-like event. It's a binary event where once you commit, you fully commit and the cell is dead within minutes. On the other hand, an unprimed cell also endures death signaling. So it's pushed towards the edge, but it's far from the edge, so it's able to survive and repair itself, or maybe not, whatever. It lives to fight another day. So maybe the difference between chemosensitivity or not has to do with whether there's a difference in mitochondrial priming. And I hope I've convinced you with BH3 profiling, we now have a tool to measure mitochondrial priming. So what I'm going to show you, I'm going to build up to some comparisons between mitochondrial priming and chemosensitivity very broadly across cancers. So this was work done by Trina Nee Keneally, now at the RCSI in Ireland, and Chris Rosik, now at Harvard School of Public Health, running their own labs. They ran this study where they looked at pre-treatment samples from patients with a variety of different diseases, in this case multiple myeloma, acute lymphocytic leukemia, and ovarian cancer, and then compared patients who eventually responded well to those who responded poorly. And they found quite consistently the x-axis, again, is an index of priming, where the higher you are, the more primed you are. And what he found is the ones who responded well, what they found was the ones who responded well were consistently more primed for apoptosis via BH3 profiling than the poor responders. Childhood ALL, a uniformly very sensitive disease, very highly primed. Endometrial, serous borderline renal cancers, very resistant to chemo, as well as most of our normal tissues, very poorly primed. So at least we could see that there is an index that is consistent with the hypothesis that I put before you. Trang Vo, a graduate student in my lab, studied this in more detail in acute myelogenous leukemia. And what she found is that she could do BH3 profiling. Again, the y-axis here is an index of priming. And if she compared patients who got chemotherapy and were cured, chemotherapy who underwent a remission and relapse, and those who were primary refractory and didn't even respond, she found that there was a gradient of priming where the ones who were cured had the highest priming and the ones that weren't cured had the lowest priming. And you could see that you could index this off of normal hematopoietic stem cells, and basically to be curable, you had to have a disease that was more highly primed than a normal hematopoietic stem cell. If we look at solid tumors, in this case ovarian cancer, this is work largely done by Chris Sorosik. He found very similar that you could discriminate patients who responded well to chemotherapy and those who responded poorly to chemotherapy based on standard normalization of CA-125, an ovarian tumor marker. You could find that a difference in priming was very good at predicting those who had a durable and those who had a less durable response to this type of chemotherapy. Similarly, Trang found that durability of response to chemotherapy was better in the patients with high priming compared to those who had low priming. And Matt Davids did very similar work in chronic lymphocytic leukemia, where those who responded better to chemotherapy were those who were more primed. So at least in these examples, it does appear that there is a consistent correlation between actual clinical response and how primed the tumor was before it even saw chemotherapy. So why are solid tumor histologies in general worse than liquid tumors? So this again, I'm going to turn to work done by Chris and Cameron now, which they were kind enough to share with me. So if you look across, this is just overall survival across all these different tumors. And you can see that it varies according to histology. And the ones who have the best overall survival, and this is all in a treated state, this is modern cancer treatment, so all these people receive some sort of chemotherapy. The ones who do best are the hematopoietic diseases, the leukemias and lymphomas, as well as testicular cancer, kind of one exception. Then the ones that do less good are the solid tumors, whether they be this sort of carcinomas or sarcomas or mesotheliomas. They definitely do worse. It's a very clear pattern that liquid tumors do better, solid tumors do worse. I probably don't need to convince you of that too hard. If you look across there are published online databases. This is the DEP map produced by the Broad Institute. If you study this according to histology, you'll again find, if you look at all cytotoxic agents put together, the most sensitive cell lines very consistently are those that involve the hematopoietic cells. And the ones that are least sensitive, this matches pretty well the clinical experience in people. The cell line experience is very similar. Across 150 different agents, you found the same thing. These cancers are most sensitive. Is this just due to these particular cytotoxic conventional chemotherapies? No. Really, no matter what therapy you throw at them, and here's all the different classes of therapies you can look at, where red indicates a higher response and blue an inferior response, you could see it's the same old pattern. There isn't just some one signaling pathway that is like one kinase pathway that is being targeted. It's no matter what you use, there's something about these hematopoietic cells that just makes them easier to kill. They die easier no matter what you throw at them. So why is that? So Chris and Cam engaged with a number of these cell lines and simply did BH3 profiling. And what they found, which is consistent with what I showed you already, is that very consistently, the most chemosensitive cell lines, the blood cancer cell lines, were also the ones that were most primed. So this is them grouped by individual cell line, color-coded, and here they are grouped according to the actual histology, and it's the same pattern. And if you actually test a correlation between the chemosensitivity using a growth index GI50 and the cytochrome C release to priming, you could see there's actually a pretty good correlation. And this is like a very broad study across all different kinds of cancers. So while I don't think that apoptotic priming is the only thing that explains chemosensitivity, I think it is probably the principle that is most important in the distinction between the chemosensitivity of blood cancers and the relative resistance of non-blood cancers. So what sets, you might ask yourself, well, what is it that sets the sort of apoptotic set point of a pancreatic cancer versus a leukemia? Why is that? Well, it has to do with the set point of the normal cell of origin. And so what Chris and Cam did, here they have the cancers ranked by where they started out. And what they, the cancers ranked by how primed they were. This is in the absence of chemotherapy, how primed they were for apoptosis. Then what they did is they took the closest normal cell they could find and did BH3 profiling on those normal cells. And what they found is that there was a pretty good tracking across all the histologies that the cells that had cancers that were the least primed arose from tissues that were the least primed. And those cancers that were most primed arose from tissues that were the most primed. So what it appears happens is that by molecular events that we don't fully understand, we understand them a little bit, and if you want to, we can discuss that later. But by methods we don't fully understand, there are some cells in our body, normal cells, that are very highly primed, and when they get cancer, they end up with tumors that are very highly primed, and also tumors that are more curable with chemotherapy. This is just a test of the correlation between the priming of the tissue of origin, the normal tissue of origin, the priming of the cancer cell. And you can see it's actually a very tight correlation, which I find pretty impressive given the breadth of the study that they did across so many different tissues. So I want to get to this Hanahan and Weinberg. This is the original basic hallmarks of cancer, which people like to refer to an awful lot. There's this idea of resisting cell death. Now, if you go back to their original paper, they were a little vague about what they meant by resisting cell death. I think it does make sense that in order to become a cancer cell, you're breaking a lot of rules that provoke death signals. Okay, so you somehow have to resist cell death as a cancer cell to eventually become a true tumor, because there are methods in place to sort of eliminate you via apoptosis. So they have to get around that. But unfortunately, I think this has been, and I've read way too many reviews that refer to cancer cells as being inherently resistant to apoptosis. And I think the answer is that, like, well, maybe cancer cells are more resistant to apoptosis than we would like them to be, but they are not more resistant to apoptosis than normal cells are. Sure, we can't cure them all, but the basic rule, the only reason chemotherapy has ever worked for the last 40 years is because there is a built-in natural therapeutic index whereby cancer cells are generally more primed for apoptosis than normal cells are. So why is relapse disease more broadly chemoresistant? If you've been following what I say, you may have a guess where I'm going with this, but I just want to point out this is like the major problem in in treatment of patients with cancer. I mean, the reason why patients die when they relapse from AML, say they get induction therapy with typically an anthracycline and cytarabine, something like that. When they relapse, the reason why they die of AML isn't only because they're no longer sensitive to that induction regimen, it's because they're not really that sensitive to almost anything we can give them. There's a reduction in sensitivity with each subsequent line of therapy that we give them, and pretty much no matter what we give them. So one thing we wanted to test, is it possible that there is a selection for a reduction in apoptotic priming that occurs with relapse? So here's an example of a small set of patients that Trang Vo studied. Some of them were sensitive, that means they underwent a complete remission and then relapsed, whereas some of them were refractory to therapy. And I think the comparison is informative. In the ones that underwent a complete remission and then relapsed, when we compared the pretreatment samples here and then the relapse samples here, there's a consistent selection for a reduction in priming. That is to say, the process of going through a remission and then eventually reemerging, the cells that reemerge have been subjected to a very strong selection pressure, and they select for a loss in apoptotic priming. However, if you looked at cells that were primary refractory, that is, they weren't even sensitive to chemo in the first place, you could see that the same kind of bottleneck doesn't occur. The same type of selection pressure wasn't applied, and there isn't selection for a decrease in priming if you look at the pre-treatment sample and a post-treatment sample. In fact, if anything, the priming might be increased a little bit. This is with conventional chemotherapy. We did a similar study in patients treated in AML with venetoclax, which is a more modern therapy. And we got the same result. Patients who were treated with venetoclax-based therapies who then relapsed, we found the same fairly consistent selection for a decrease in apoptotic priming at the mitochondrial level. This is work done by Jeremy Ryan and Thelma Mashaka, technicians in the laboratory. So Shruti Bhatt attacked this problem even more broadly. So Shruti she's now at the National University of Singapore, but was a postdoc in my lab. Shruti studied a large set of AML PDX models. These are patient-derived xenograft models where you take an actual human AML, put it in an immunodeficient mouse, and grow it up in that mouse. And in a very painstaking process, she took several of these models and exposed them to different drugs to select for resistance. So the drugs she used were quizartinib, which is a kinase inhibitor, birinapant, which is a SMAC mimetic, and venetoclax, which is a BCL2 inhibitor. And she selected for resistance over months. So this is a very painstaking experiment. And then what she did is she had a model for this one. You can see across the top, the PDX model called DFAM61786, for instance. She had one model that was the parental model, then one that was a quizartinib resistant model, one was a birinapant resistant model, one was a venetoclax resistant model. So very, very different drugs. And then she performed BH3 profiling to measure the priming of the parental and compare it to the resistant models. And you could see, without going through every single one of those bars there, because there's a lot of them, the black bars are bigger than all the other colored bars. And what that means is that the priming of the parental sample is consistently higher than that of the resistant samples. That is to say, whether the drug is quizartinib, birinapant, or venetoclax, there is a consistent selection for a reduction in mitochondrial priming. So now, to explain what Shruti did next, I want to introduce you to dynamic BH3 profiling. This is a little different. It uses BH3 profiling, but this asks a slightly different question. It asks, if we apply a drug to a cell, does the priming increase? So it's the same thing as BH3 profiling, but there's a drug pretreatment, and we compare essentially a drug-treated well with a non-drug treated well and ask if there's an increase in priming in the treated well. And it turns out that that's a good predictor of a drug that's really effective in vivo if you increase the priming in this ex vivo assay. And here's an example of the process. This is for mesothelioma, but it's similar for whatever tumor you have. You take the tumor, make a single cell suspension. If it's a leukemia, you don't have to do that, obviously. You distribute to a 384 well plate. You treat it with drugs. Our treatments are generally less than 24 hours. And then you do the BH3 profiling and compare a treated well with a drug-treated well. And where we see an enhancement of priming, that is to say, an enhancement of mitochondrial sensitivity to whatever our test peptide is, usually the BIM peptide, that's what we call delta priming. And if we observe delta priming caused by a drug, that generally means it's a good drug. So what Shruti did is she asked the question, could she actually predict sensitivity of her PDX models to different drugs based on this dynamic BH3 profiling approach? This is still unpublished, hopefully soon to be published, but we will see. And what she did is she simply did dynamic BH3 profiling across a lot of different samples. Then she treated these models in vivo with drugs and had positive negative controls. That is to say, drugs that were expected to work in this model via dynamic BH3 profiling, and drugs that were expected not to work in this model. And so she tested a number of models and a number of different drugs, and these are the results that she found. So this is the kind of broad dynamic BH3 profiling. I'll point out something that's very interesting, which is just as you observe in the clinic, those PDX models that were derived from patients that were treatment naive are more broadly sensitive to almost all the drugs she used, and here's the couple dozen drugs she used, whereas those that were determined for relapse patients here under the yellow bar were almost uniformly less sensitive to everything we treated them with. So she was able to replicate a very important clinical phenomenon with this. So then she took a selection of these and actually treated the models and asked how well can she predict response. So what you see on the x-axis in all these is the delta priming value. That's our assay readout. And on the y-axis is an index of actual chemosensitivity in vivo. And you could see across all these drugs that are very different, birinapant, JQ1, an epigenetic modifying agent, venetoclax, quizartinib, consistently BH3 profiling, or dynamic BH3 profiling, was a very good predictor of actual in vivo activity. And I will say that we don't really have any other great predictive biomarker for any of these, except for maybe quizartinib, so that in these cases, it would be beneficial for the treatment of probably all of these drugs to use a functional predictive biomarker to stratify patients in their use. This is an example of a human clinical trial in AML. I'm focusing here on leukemias because I think there's more of an interest in leukemia here at the center. But um, uh, in this case, Jacqueline Garcia had this clinical trial at Stanford, and there was a related one at Mass General Hospital. She combined those patients. These are patients with AML who were treated with the reinduction regimen of lenalidomide plus MEC, which is mitoxantrone, etoposide, and cytarabine. And Shruti did an experiment with dynamic BH3 profiling. She was totally blinded to the outcome. She didn't know what the outcome was going to be. And she was able to rank patients based on their dynamic BH3 profiling and then unblind and compare how well that predicted complete remission. And you could see it did a fantastic job. She was able to predict with nearly, not completely, but with nearly 100% accuracy, those patients who would undergo a remission to this Len-MEC regimen and those who did not. And this is what the receiver operating curve looks like. This is a receiver operating curve is used to compare the efficiency of binary predictive biomarkers, where you have sensitivity on one axis and specificity on another. And you can see that this performed extremely well, and this area under the curve of 0.98 would compare very favorably to any clinical biomarker used nowadays in clinical cancer medicine. So getting back to Shruti's models, I told you these are the models I showed you before, where you can see the parental model is more primed than all of the resistance models, no matter what. So she took all of these resistance models and then did dynamic BH3 profiling on them to determine whether there was any loss in chemosensitivity in these different models. And these results are represented in a pie chart. The parental model, where red indicates drugs where there was sensitivity and blue where there wasn't sensitivity. And here you can see in the parental model, basically, the main point is the red is bigger in the parental model across the board. And this, I think, teaches us the lesson that when you select for resistance to any drug, you are probably selecting for a reduction in apoptotic priming, and that corresponds to a simultaneous broad loss of chemosensitivity that she painstakingly did in her PDX models, but I think this happens every single day in the clinic. So when we consider all these questions, and a question I didn't address, which is the superior performance of young people with ALL compared to old people with ALL, and yes, the priming is different between those two different diseases. I think apoptotic priming probably explains a lot of these big questions. It's not the only principle, but I don't know of any other concept or any other principle that can answer these very large questions that we take for granted. We take them so much for granted in clinical oncology, we don't even ask the questions anymore, but I think this is probably the biological basis for these. So what can we do about it? If it's bad to have a cell that is poorly primed for apoptosis, well, maybe we can increase it in cancer cells. So one way to do this might be by identifying the right BH3 mimetic. BH3 mimetics are a new class of drug that inhibit anti-apoptotic proteins like BCL2, BCLXL, and MCL1. So maybe if we choose the right BH3 mimetic, it'll prime cells for apoptosis and make them more chemosensitive. So now I'll turn, remember this chart I showed you, up till now we've just been using these promiscuous peptides to measure overall priming. There's these selective peptides that can do a different job. Notice this HRK peptide, for instance. This selectively interacts only with the BCLXL anti-apoptotic protein. So if you have a mitochondrion that's sensitive to the HRK peptide, this is a mitochondrion in a cell that's very BCLXL dependent. If you're sensitive to the NOXA peptide or another peptide we use called MS1, if you're sensitive to the NOXA or the MS1 peptide, then you're very MCL1 dependent. And if you're sensitive to the bad peptide, but not to the HRK peptide, that probably means you're very BCL2 dependent. And that's important because we have different drugs that target BCL2 from MCL1 from BCLXL. So it looks like we could use BH3 profiling to discriminate BCL2 and MCL1 dependent cell lines or leukemias. Could we really do this? So these are actual leukemias from mice, and these are kind of designer leukemias. We made one leukemia. These are both, if you will, BALLs. This is a classic model. The BCL2-MYC model was invented by others from the Weihai Institute in Australia far before me, but I exploited it just for this test. So we made this BCL2-MYC leukemia, and then we made one, and that's explicitly BCL2 dependent, and then we made one just like it, an MCL1-MYC leukemia that's MCL1 dependent, and if our method works, we should be able to discriminate these two, and in fact, we could exactly as we predicted, where you can see the MCL1 dependent leukemia, its mitochondria are sensitive to the noxapeptide, but not the bad peptide, and exactly the opposite for the BCL2 dependent leukemia, which is sensitive to the bad peptide, but not the noxapeptide. So what we have now, we actually do have a test that can identify BCL2 dependence and MCL1 dependence. So we got this result over 10 years ago, and what Vicki Moore in my laboratory did is she turned her attention to real human leukemias because we knew BCL2 inhibitors were coming, but they weren't quite there yet. She turned her attention to real leukemias and asked, can we identify the ones that are BCL2 dependent? And she turned her attention first to CLL. And what she found, and this is, I like to say, one of the most important bar graphs in my career, what she found is that uniformly, and very homogeneously, CLL patients had diseases that were sensitive to the bad peptide, but not the noxapeptide or the HRK peptide, meaning that CLL was very broadly, very consistently a BCL2 dependent disease. And this was before there were clinical trials of venetoclax in CLL. But her prediction, as well as other data, spurred the initial clinical trials of venetoclax in CLL. Venetoclax is a drug here called ABT-199, and it's a selective small molecule inhibitor of the function of the BCL2 protein. So this was first tested clinically in CLL, and this is a result of one of the early clinical trials led by John Seymour. He was the first author in the New England Journal of Medicine. You can see that indeed, as expected, venetoclax is very broadly effective in CLL, just as Vicki predicted. In fact, if you give venetoclax or venetoclax with rituximab, there's something like an 85% response rate in patients with CLL. So her prediction using purely functional means was completely vindicated, I think, by this clinical trial. Very similarly, Trang Vo turned her attention to AML. Now, this was a disease that no one had any suspicion would be BCL2 dependent because BCL2 was, for no very good reason, thought to be a lymphoid protein, and myeloid cells would depend on other proteins. But it turns out, actually, if you do the empiric work that Trang did, that most samples, those from patients that were sensitive to chemotherapy, that is, underwent a CR, and those that didn't undergo a CR, most patients, but not all, with AML have an AML that is BCL2 dependent, or at least more BCL2 dependent than our comparator HSC cells. So using this data, as well as some functional data produced by a collaborator, Marina Konopleva, then at MD Anderson, we took this data to AbbVie and convinced them to start a myeloid program that most people had been reluctant to start until then. And this turned eventually into the first clinical trial that was published, I guess that would be 7 years ago. And this is just single agent venetoclax used to treat patients, most of whom had relapsed refractory AML. And we thought it was pretty amazing, even in this fairly dire situation, you could see there's definite biological activity in most of the patients, including some complete remissions. That's what the green bars are. This data was good enough for AbbVie to expand its trial to a trial very commonsensically just combining venetoclax with agents that we knew already were effective in AML, like azacitidine, hypomethylating agents. And this is the so-called VIALE-A trial that was published a couple years ago that showed a clear benefit of adding venetoclax to azacitidine. If you look at complete remission rates, you can see that whereas azacitidine has a complete remission rate in the 25% range, the azacitidine plus venetoclax combination had a complete remission rate in the 70% range. So dramatic increase based on venetoclax. And I just want to point out, this was all identified, this was precision or medicine, whatever you want to call it, all done in a perfectly functional way. There are no BCL2 mutations, copy number variations in AML or CLL that would have told you it would work in these diseases. And there's no way that genomics could have done the job that pure, simple functional tests were able to do. So nowadays, a very similar patient to that patient I talked to you about before. I'm no longer an active clinician, but my colleague Jacqueline Garcia is. This is an example of one of her patients. 75-year-old AML patient comes to her clinic, no longer has to be sent home, told she's going to die in a couple months. She can get venetoclax and Vidasa, stay home most of the time. It's not a long in-hospital stay for the induction regimen, and she has to continue to take her maintenance therapy, but 18 months later, this patient is still in a complete remission. So a big increase, I would say, an important improvement in options that are available now for older patients with AML, which is most of the patients with AML. In just the last couple of minutes of my talk, I think I want to talk about immune cells because I think all of us are interested in how in immunotherapy, and I think justifiably so. We've seen some amazing results where literally cell-based therapies are able to cure people of cancers that were about to kill them. Now, this has occurred mainly in ALL, probably also in lymphoma. It's been harder in solid tumors. I'm not going to get into why because I don't know why, but I suspect priming has a lot to do with it. And that's because I sort of asked the provocative question, do immune cells kill cancer cells via magic? And of course they don't, but you might think so to like read a lot of what is written about immune cell killing. And that's because immunologists like to study immune cells, and they like to study, in the case of CAR T cells, like making these CAR T cells, making them better, making them expand better, making them faster, making them persist. And those are all important. Don't get me wrong. That is very important work to do. But in general, there is less study of what happens after the T cell, the CAR T, recognizes its target cell. And if you have a T cell that engages its target cell, but you don't wind up with a dead target cell, it doesn't matter that you got the T cell there. You have to end up with a dead target cell for this to actually be therapeutically beneficial. So it's important to understand how T cells, or whatever cytotoxic cell you're using, actually ends up killing the target cell. And I should tell you, it's via programmed cell death pathways. This is not something I'm inventing. This is well known for a long time that whether it's NK cells, and I'm going to show you studies with NK cells, but whether it's NK cells or T cells, the programmed cell death pathways are very important, whether it is via ligation of death receptors with like TNF or Fas ligands, or the mitochondrial pathway of apoptosis, or non-apoptotic mechanism. Programmed cell death pathways are important. But I don't have time to show you all the work that Aaron Pan did on this. This was published last year, and you can look it up. But he used NK cells, and he spent a lot of time showing that in his models, it was killing via the mitochondrial pathway of apoptosis. He did things to block apoptosis, and they no longer killed as efficiently. And he blocked all the other programmed cell death pathways, and it had little or no effect. So I just want to, you would have to go through a lot of data to see it. It's all in the paper. But in his models, we are seeing apoptotic killing by NK cells, which is probably the main mechanism of killing in vivo as well. And again, not totally invented by us, but he just validated in the systems he was using. And so what he wanted to ask the question is, if we prime cells for apoptosis, can we then make them more sensitive to cell-based therapies and make cell-based therapies work better? Because there's a lot of room improvement in cell-based therapies, even though we're curing some people. And so he turned to BH3 mimetics. And he wanted to, in his two models he chose, one was HeLa cells and the other one was a leukemia. He wanted to know, could he pick the right BH3 mimetic? And if he picked the right BH3 mimetic, would it actually make NK cells work better? So he chose BH3 mimetics by the method I told you we can do it, and that is BH3 profiling. And what he found is that this HL60 cell, engineered to overexpress BCL2, no surprise, gives us a BH3 profile that is a BCL2 dependent signature, that is sensitivity to the bad peptide, but not the MS1 peptide, which is just like the Noxa peptide. And HeLa is exactly the opposite, where HeLa is sensitive to the MS1 peptide, indicating MCL1 dependence, but not to the bad peptide, so there's no BCL2 dependence. So you would give an MCL1 inhibitor to the HeLa cells, we would predict, and a BCL2 inhibitor to the leukemia cells. One's a solid tumor, one's a liquid tumor. So how did that work? So he constructed an in vivo model, basically injecting immunodeficient mice first with the cancer, and I'll show you the leukemia first, and then after some days, after the cancer was established, injecting NK cells in the presence or absence of these BH3 mimetics. And he used both the positive control, the one we predicted would work, and the one we predicted would not work. And what you can see here, this is tumor volume on the y-axis here, is that there's only one that really worked well, and that was NK cells plus the BCL2 inhibitor. If you use the drug alone, it didn't work very effectively. If you use the NK cells alone, it didn't work very effectively. Maybe some modest effect, but used together, they work very well. And there was no benefit to adding the MCL1 inhibitor, just as we had predicted from our BH3 profiling. And this is whether you look at tumor volume or if you look at survival to the right, you get the same answer. In a very nice comparison, we used the HeLa cells, which were predicted to be sensitive to the MCL1 inhibitor. And what you find is the only treatment that really works well is the NK cells plus the MCL1 inhibitor. If you combine it with the BCL2 inhibitor, there's very little effect and no different from the NK cell at all. And that is whether you look at the tumor volume in vivo or you measure the tumors having been excised from the mouse. So it appears, and I think this is probably, he studied in this narrow context of NK cell killing. But I think this probably generalizes to immunotherapy more broadly, and we're studying it more broadly. And that is, maybe we don't have to be too sophisticated in how we're going to assemble combinations for immunotherapy. And this probably extends to checkpoint blockade, which ultimately relies on cytotoxic T cell killing too. Maybe what we need to do is just focus on identifying drugs that can prime the target cell for apoptosis. And probably any drug that can do that is going to be able to accentuate or make much more efficient the cell-based killing that we're relying on for clinical response. So just to summarize, and I'm about to conclude, I think apoptotic priming probably explains a lot of what we've observed in clinical oncology without really knowing it. And that we can use some very simple understanding of these principles to do a better job to maybe more quickly assemble a combination of drugs. And this includes not only drug-drug combinations, but also immuno-oncology and drug combinations. So I want to thank the people who did this work. I tried to thank them as I go through, but they're listed here in front of you. And I'd be very happy at this point to take any questions.
Thank you very much, Tony. It was great for giving time to the attendees. So, and I think it was great and completely original. I like it very much. If you, the priming to apoptosis, at one point you said genomics doesn't matter so much. So there's no genomics at all that can predict priming to apoptosis? There is something in the genomics that can predict or not? Or is the cell types like you did in these graphs?
Okay, so I think when I said genome, I was specifically talking about
you talk about one aspect.
Yeah, venetoclax activity, AML, CLL, genomes can tell. So you certainly can do genetic manipulation of a cell line and do things to make it more primed or less primed. For instance, if you take a cell line and overexpress BCL2 in it, it's going to get less primed. Okay, or if you knock out BCL2, it's going to become more primed. So it's not to say that genetics has no impact on it. So what you might ask is, like in AML, have we been able to detect genomic signatures of more or less primed patients? Not really. And the problem, I think you probably could if you had enough patients, but as you know, one of the problems is there's too much heterogeneity in the genomics of AML, so that you would need a very large number of patients to be able to make statistically significant comments about any particular genetic alteration. I suspect it is the case. And like I said, we've done a lot of different, and you can also do, Chris has done similar work with oncogenes, and oncogenes can affect the priming for sure. I think it probably is the truth that genetics can affect priming. We haven't been able to study it in actual patient samples due to the difficulty in assembling a large enough data set.
For example, I was thinking, those tumors with microsteroid instability maybe have more priming. No? They're more prone to die by apoptosis. For example, we have those old DNA damage everywhere there.
It could be, but I don't know the answer to that. So I would say in order to answer that, a disease with a lot of microsatellite instability like colon cancer, very hard to get colon cancer samples to do that sort of study, but it's a reasonable hypothesis.
Thank you very much. Questions?
You just mentioned the genomic heterogeneity of cancers. So how much heterogeneity do you observe in terms of priming in a cancer?
Right. So Everything I showed you here, I think I think all the cases I was showing you kind of the population-based summary or the bulk summary, but all that involved single cell information. So we actually know for every population we've looked at, whether it's a patient tumor or it's a cell line in vitro, there's always a distribution of priming. Okay, so all I can tell if there's definite heterogeneity, and you'd have to look at the different distributions. They all have their different coefficient of variation and so forth. What is interesting, though, is if you take the best predictor, for instance, in response to AML, is if you take advantage of this heterogeneity and you ask yourself, how primed is the least primed 5% of the cells? That turns out to be the best predictor of response. So I do think that heterogeneity plays an important role, and it is the least primed cells that are the hardest to eliminate that are going to dictate curability.
Hi, Tony. Very nice. Thank you. So you nicely showed that the priming determines the response of cancer cells to chemotherapy and targeted agents, but is this the same for the BH3 mimetic like venetoclax or MC1 inhibitors? So is there a nice correlation between priming and response to these molecules? And does it make sense to use that in the clinical to determine which patients will benefit?
Right. So I can only answer that for venetoclax because there haven't been enough people treated with other BH3 mimetics, or at least not that I've been able to get my hands on. So for venetoclax, BH3 profiling can predict response, but not as immediately as I would have guessed. So the immediate BCL2 dependence, you might think that, as I thought, would be the best predictor. And in fact, it isn't. What is the best predictor of response is the absence of MCL1 or BCL-XL dependence. So clinical response is evaluated at 30 or 60 days. So I think what happens is that's plenty of time if there are adaptive mechanisms like turning up MCL1 or turning up BCL-XL or one of the other anti-apoptotics. If that's already available to the cell, they're less likely to have a clinical response. And I think that's what we're detecting. So our prediction of venetoclax sensitivity has to do with the absence of MCL1 and BCL-XL dependence.
Okay, thank you.
We have also questions, a question from the online people.
Okay.
Here. So what about radiotherapy? Also predicts priming response to radiotherapy?
Radiotherapy, yeah. So so Chris Sarosik has actually done some work largely in normal tissues rather than cancer tissues. But yeah, there's like the most radiosensitive tissues, like lymphoid tissues, are much more primed than radio-resistant tissues. So I think yes, the main predictions of sensitive radiotherapy are also going to be apoptotic priming.
Well, first of all, Tony, congrats on the near 20th anniversary of the lab, which I witnessed its emergence, and I think you guys have to be really happy about the contributions to the clinic you've made now, taking this perspective.
Thanks.
So the chemotherapy question in terms of prediction in the clinic, I don't know if you have ever thought on how to implement this. I mean, if it's all about priming with chemotherapy, have you thought about implementing it in some way that you can spare treatments to people who might respond?
Yeah, and it's been a challenge. Like I'll give you an example, ovarian cancer, which you study. So one of the problems in ovarian cancer has been, I think we have a way to predict. So what happens in ovarian cancer, for those who don't know, often what happens is a patient has a resection, and then they'll get carboplatin and Taxol after the resection to clean them up. Clean up the tumor bed and hopefully have a response. And I think we have a way of predicting who's going to do well with that therapy and who won't do well with that therapy. So I talked to our ovarian cancer oncologist and I said, what would you give a patient who I didn't think was going to be as sensitive to carboplatin and Taxol? And they said, we would give them carboplatin and Taxol. And that's because, and that was really woke me up to the idea that it's not enough to just show that you're going to be sensitive or not to a particular therapy. You have to provide an alternative. And one of the challenges of showing poor priming is that what you're saying is they're probably going to respond poorly to everything. And that's why in terms of assigning patients to therapies, we're turning our attention more to dynamic BH3 profiling, which not only tells you whether you'll respond to this drug, but tells you, okay, if you don't respond to that drug, there's these other alternatives that you might respond to.
So related to that, and you might remember we entertained the idea of doing some profiling of the CCLE, the Cancer Cell Encyclopedia, to see if we could predict with genomics. Do you think that would still be a good idea, or for solid tumor deployment of these kind of therapies, or don't you think don't think that's going to be informative?
To do that, I'd connect you to Chris, because as you may have seen, like he took a whole bunch of cell lines. I think he just restricted his attention to the NCI 60, but but you could do the same thing for that. And he tested the priming in all of those, and that would be, and the genomics of that is readily available. I don't know that he is exploring that or not, but if I'd be happy to put you in touch with
numbers might be important there, 60 versus 1,000 or something,
right.
But logistically and funding wise, it's complicated. It's complex.
True. I think you get more information out of a 1,000 cell line database, and you're welcome to ask him if he wants to do 1,000 BH3 profiles.
Convince somebody to pay for that.
Yeah.
So last question is about organoids. Like, have you thought or have you tested like BH3 profiling on these more complex model systems, and do you think the information would be different?
Yeah, in fact, we've done it in ovarian cancer organoids, where we used similarly to see if we could predict carboplatin sensitivity, and basically it works. So that's not that we haven't published that yet, but I mentioned I'd be happy to connect you to one of my postdocs, and that's the person I'd want to connect you to. We do it by imaging, so the imaging is a little more tricky because it's not a flat monolayer, but she was still able to figure out a way to do it.
Yes, thanks. Very nice talk. In the case of chronic lymphocytic leukemia, we have seen exceptional cases in which after years of follow-up without treatment, the disease disappeared. So I wonder that there is an underexpression of the anti-death protein in these cases, and I would like to know your opinion about.
I guess I don't have any information to add to that. That's a rare enough event that we have never studied like a cohort of patients who have these very long-term remissions. And I think it's a plausible hypothesis, and it makes sense. Maybe those were like the most primed cases, but we simply don't know that yet because it's too rare an event.
Okay, thanks.
Thank you very much. Just in the interest of time, we should finish here. Thank you very much again.